The brain grows from the inside out, with the radial glial cells whose projections are shown here serving as a scaffold for migrating cells. Remember once again that the cells of the neuroepithelium span the entire apical basal surface and that this is one cell layer thick, but that the nuclei are found at different heights. The reason why these cell bodies appear to be at different heights is because they actually migrate during the cell cycle through a process that's known as interkinetic nuclear migration. And so what happens is that you can see here in this figure that when the cells are in S phase, their nuclei are located near the basal end. So this is basal. So this is outer surface, and the apical surface is on the other side. And when the cells are undergoing mitosis, you can see that the nuclei are located near the apical end, so near the lumen of the neural tube. Once the cells enter into G1, they begin to move basally once again. And this is something that not only happens in neuroepithelial cells, it also happens in the radial glial cells. And you can see this process of interkinetic nuclear migration happening in these figures. This is in the neuroepithelium. And you can see that when the cell is in mitosis, because this cell is dividing here into two cells, it is closer to the apical surface, and as the cell enters G1, it begins to move basally. And this is thought to be related in some way to microtubules and motor proteins, because when you have mutations in those, this process doesn't happen properly. However, that could just be the fact that you need microtubules to function properly in order for cells to form the mitotic spindle and to divide. And as this process of interkinetic nuclear migration is happening, there's another process that I didn't show you yet, but it's related to the asymmetric division of these neural stem cells. So the stem cells have the capacity to divide, if you recall, both symmetrically and asymmetrically, and they can divide symmetrically to either give rise to two stem cells or two committed cells. And remember that if all of the stem cells are dividing symmetrically to give rise to stem cells, then the stem cell pool expands. If they're all dividing symmetrically to give rise to committed cells, then the stem cell pool shrinks. However, there could be population asymmetry, so that some stem cells are dividing into stem cells and others are dividing into committed cells. So they're actually maintaining the stem cell pool while also giving rise to committed cells. In the case of the neuroepithelium, they initially divide symmetrically to give rise to stem cells, but then they begin to divide asymmetrically to give rise to a stem cell and a committed cell. When the cells divide asymmetrically, which is actually what happened here, you can see that one of the cells remains associated with the apical end. It's worth noting that it's also connected at the basal end, and this cell will remain as the stem cell, whereas the other cell actually detached from the apical end, and it now looks a little different. So the staining that you see there in yellow is a staining that's specific for neurons. So this cell went on to differentiate, while the other cell, the one that remained attached, remained as a stem cell. So as the neural tube matures during the process before neurogenesis begins, these neuroepithelial cells are going to give rise to the ventricular radial glia. And these ventricular radial glia are going to undergo both symmetrical and asymmetrical divisions to maintain the population and also give rise to more differentiated cells. Now, an important thing to note is that you see that the ventricular radial glia remain associated with the apical end. And this region where you have the ventricular radial glia is known as the ventricular zone. In addition to that, you have a second zone that's known as the outer subventricular zone, where you have cells that derived from this radial glia, and these include the outer radial glia, which is shown here in the yellow, and the intermediate progenitor, which is shown in the orange. And these two cell types, the outer radial glia and the intermediate progenitor, are going to be cells that are going to give rise to different neurons as well. And I'm going to talk about what the differences are between the three cell types in a moment. But an important thing to know is that early in development, you tend to have more symmetrical divisions to give rise to stem cells. And as development progresses, the number of asymmetric division increases so that you can maintain a stem cell, but also begin to expand the number of differentiated cells. And these cells that are forming in the outer ventricular zone or outer subventricular zone, and the ventricular zone are going to be giving rise to all the different cells that form the cortex of the brain. The cells of the germinal zone, which is a zone that includes the outer subventricular zone and the ventricular zone, are going to give rise to all the cell types of the cortex of the brain. And they include three major cell types. Those three major types are the ventricular radial glia, which you can see are associated with both the apical and the basal end. We have the outer radial glia, which are only associated with the basal end. That's why they're known as outer radial glia. And we have the intermediate progenitors, or IP. And the intermediate progenitors, these are like the transit amplifying cells. And so it is thought that the potency of these cells, so the number of cell types that they can give rise, is such so that the ventricular radial glia have the greatest potency, followed by the outer radial glia and the intermediate progenitor. And that's true for both the potency and also for the self-renewal capacity. So the ventricular radial glia are the ones that have the greatest self-renewal capacity and the greatest potency, or the potential to give rise to the most or greatest number of cell types. And it's important to note that both the outer radial glia, and let me use a different color here, the outer radial glia and the intermediate progenitor come from the ventricular radial glia. And then the intermediate progenitor can be derived from the outer radial glia or the ventricular radial glia. So this is where we have this distinction where the ventricular radial glia have the greatest potency and the greatest self-renewal capacity, followed by the outer radial glia, and finally the intermediate progenitors. Different types of neurons and glial cells are born at different times during development. Labeling these cells at different times has shown that the cells that have the earliest birthdays, so these are the older cells, are closer to the ventricle. So these would be older. So these cells are migrating the shortest distances, whereas the cells that have the later birthdays, the younger cells, are migrating the farthest distances, and these are located closer to the outer surface of the cortex. The differentiation of those cells is going to depend on the positions that the neurons occupy once they're outside of the germinal epithelium of the ventricular and subventricular zone. The mechanism for the formation of these different layers involves the movement of these cells along the process of the radial glia, so using their sister cell to migrate to the correct layer. In addition to that, remember that cells that have the earlier birthdays are going to populate the layers that are closer to the ventricular zone, whereas cells that are born later are going to populate the outer layers, the cells that are farther away from the ventricular zone. And we can see this here as well if we label these cells at embryonic day 29 in the ferret, which is when the cells are supposed to be migrating. We see that they migrate to layer 6, which is more interior, closer to the ventricular zone. However, if we were to label these cells at postnatal day 1, at this point, the cells are migrating to layers 2 and 3. So the brain is growing from the inside out, with the inner layers forming first and the outer layers forming above those layers later. Another important aspect of this is that the fates of the cell become more restricted as development progresses. So those cells that are born earlier have the potential to give rise to any neuron, whereas the later precursors can only form neurons of the upper layers. And that's related in part to the stage of the cell cycle that the cells are in when you take them, because a younger neuron has the potential to become part of an inner layer and also an outer layer, but we're not going to concern ourselves with that aspect of how this relates to the cell cycle. Whether a cellular division gives rise to two stem cells or to two different progeny is also based on the plane of cell division. If the cells divide perpendicularly, then they give rise to two stem cells, but if this plane is oblique, then one of the progeny will become a differentiated or committed cell, and the other one will remain as a stem cell. And if you were to take cells and you alter their planes of cell division, so if they're supposed to divide in perpendicularly and you make them divide with an oblique plane of cell division, you can alter the fate of these cells so that they begin to differentiate earlier than they should. In an asymmetric cell division, as you know, one cell remains as a stem cell and the other one goes on to become a committed cell. And as it turns out, the decision to become a committed cell or a stem cell is related to the centriole that the cell inherits. So if the cell inherits the older centriole, it'll remain as the stem cell. If it inherits the centriole that's the result of the duplication, so the daughter centriole, then it will become the committed cell. And the reason for that is because the older centriole is associated with the primary cilium, which is projected into the lumen of the neural tube. And in the lumen of the neural tube, you have all these factors that are important for maintaining that radioglial stem cell as a stem cell. So the daughter cell that inherits the centriole that is a daughter centriole does not have that association to the primary cilium. And because of that, that cell is no longer able to make contact with the factors that would allow it to remain as a stem cell. It will eventually form its own primary cilium, but that cilium is going to be pointing toward the basal surface.